In this video, we're going to be talking about how parasites can help us understand the lives of endangered species, extinct marsupials, and even ancient mummies. Before we get started, I have to warn you that this video might make you itchy. We're going to be talking about creepy crawlies and spooky mummies. So if that's not your thing, it might be a good idea to check out one of my other videos. With that out of the way, let me introduce you to today's expert, Gaia Mortier. Gaia is a PhD student at the University of Reading, where she studies arthropod ectoparasites with the aim of understanding past human and animal life histories. Stay with me, because if that last sentence made you scratch your head like you've got a wig full of nits, then you're closer to understanding Gaia's work than you might think. Let's kick off by asking Gaia what she means by arthropod ectoparasites.
So arthropods are animals that belong to a grouping of invertebrate animals, so they don't have a spine, and they have jointed legs, and basically their skeletons are on the outside, which means that when they grow, they have to molt their skin off. Within the arthropod, you'll find like arachnids, you'll find insects and all that kind of stuff. Ectoparasite refers to parasites that live on the external layer of their host. So a good example of that is fleas, you have lice, but also ticks are ectoparasites.
So arthropod ectoparasites are parasites, obviously, that live on the outside of their host body from the family of animals that includes insects, spiders, and something quite horrifyingly called tongue-eating lice. Moving very swiftly on, let's ask Gaia what she means by life histories.
Life histories in archaeology is usually when we look at an individual's life, and that could be a human, that could be an animal, and basically what stages in life they went through and how they did it. Ticks are quite a good example of where they very clearly take a break in their life cycle to molt and then allow for feeding. And the same with fleas, actually. They molt in a fashion where they lay eggs and then the eggs drop off, then they form larvae, and then they become adults, and then they jump on.
Now we understand that Gaia studies ectoparasites, including fleas, ticks, and lice, with the aim of understanding how and where past humans and animals have grown, survived, and reproduced. However, we still don't understand how the biochemistry of these ectoparasites helps us understand the life histories of other animals, especially of extinct species or long-dead mummies. I promise we'll get to the mummies soon, but for now, let's ask Gaia what the remains of these nasty little creatures can tell us about other animals.
If you're talking about isotope analysis, a lot of people glaze over, including me a year ago, and sometimes still, where isotopes are forms of molecules that we find out in nature. So elements have specific isotopes that are in specific ratios all across nature and then get absorbed into tissues. So that's quite a complicated system, but all you need to take away from it is that certain things such as digestion and location impact these ratios. So if we analyze, for example, my teeth, you could see what I was eating when I was, whenever my teeth fell out or regrew the new teeth. And also where I was doing that through the water that I drank and the isotopes that get absorbed in the water molecules, for example. A diet can tell you a lot about individuals and even populations.
The ratio of isotopes, or different flavours of the same element, vary between different locations and types of food. Because ectoparasites' diets are exclusively supplied by their hosts, the isotope ratio found in parasites tells us something about where their host lived and what they ate. Isotope analysis of ectoparasites is a bit like rummaging through someone's bins and working out where in England they lived based on the ratio of leftover Greggs sausage rolls to overpriced sarnies from Pret. Like isotopes with variable geographic distributions, finding out which of the two someone visits more frequently might tell you something about where they live. Isotope analysis already gives us a great insight into the life histories of past humans and animals, but is there anything else we can learn from their ectoparasites?
Every species of animal, and in some cases, unique individuals within these species have a DNA barcode or DNA sequence that we can then analyze and compare to other ones so we know what we're looking at without seeing the whole picture. You can get quite clear DNA from the host preserved by parasites, and especially when you look at eggs from lice, which are usually called nits. They have this cement-like protein that they use to glue their eggs to hair. In a lot of cases, this protein encapsulates a lot of environmental DNA, which in some cases still contains the DNA from the host. And it's like this time capsule that throughout time doesn't degrade as much as anything around it does. So for example, if you find it in hair, the hair can be quite degraded. Even if it doesn't look it, the DNA will degrade over time. Whereas if it's encapsulated by this protein, it's incredibly well preserved. So by then finally breaking open this egg and then analyzing what's inside it, we actually get better DNA results than, for example, if we were to analyze the hair or the bone of the person or animal itself.
Well-preserved host DNA could theoretically allow extinct animals like the Thylacine, an extinct carnivorous marsupial and species Gaia is keen to study, to be brought back to life. However, 3 books and at least 6 films have demonstrated how this can go horribly wrong, and Gaia cautioned that bringing back some species could do a lot more harm than good.
De-extinction is quite a tricky topic, I think, because you're talking about obviously reintroducing animals in an environment they haven't been in in some cases, a significant amount of time. How would the ecosystem, respond to them being back or respond to them being there at all? In 100 years, a lot changes, and the extinction of the Thylacine, for example, was very human-driven. So from that perspective, yes, it would probably be quite a good candidate. But in a lot of ways, it's quite a keystone species in an ecosystem because it's a large predator. And that obviously has a lot of knockdown effects, say, to the prey species below it, and also the other predators that have now adapted to them not being there.
Now, you might reasonably be asking why all of this fancy isotope and DNA analysis can't just be done on the preserved remains of the host human or animal itself. And in my opinion, this is where things get really interesting. We're finally going to talk about mummies.
And with mummies, it's often quite a significant thing because they're very culturally important, obviously, because it's the remains of ancestors. So if you look at the lice that you find on them, it would basically be an avenue through which you don't have to sample the mummy itself, and you could still learn the same things about it.
If you're not from the UK and you've ever heard about the British Museum, it's probably because of their historical disregard for the cultural significance of many artefacts. As people are becoming increasingly fed up with what I like to call Indiana Jones style archaeology, it's important to develop more respectful methods to collect important data from culturally significant artefacts. Gaia's techniques allow us to gain a better understanding of the life histories of past humans and animals without destroying their remains. This will help preserve these artefacts for future generations and could help scientific institutions like museums with their ethically debatable practices and PR problems. Clearly, host animals and humans don't have an infinite amount of ectoparasites, so I would imagine that Gaia wanted some practice with these techniques before grinding up too many ancient creepy crawlies, especially considering the obvious and highly scientific threat of deadly curses.
Up until now, I've been mostly working on a modern testbed, so that means I've been looking at samples that are no older than 10 years old to kind of see how the techniques would work on something that isn't necessarily as important as something that's historical or even ancient samples. And what we've been gathering is that parasites actually very interestingly reflect on their host on more of an average scale. So I've been looking at ticks, and ticks have quite a distinct life cycle where they feed on the host 3 times throughout their life. What that means is that they will, for example, they will feed on a host, drop off to molt, so regrow their entire exoskeleton, and then go on to feed again. Which means that if you look at a tick, if you look at the blood meal and the exoskeleton separately, they are 2 separate hosts. So we're trying now to kind of pick at that and put some puzzle pieces in place to see what that would actually mean if you just find the exoskeleton, for example, in an archaeological setting.
Gaia thinks this approach needs fine-tuning before being implemented with the more culturally sensitive artefacts we were talking about earlier. Finally, I asked Gaia what the next steps for her research will be.
For next steps, so excitingly, I've been able to actually look at a lot of Tasmanian tigers because I keep finding parasites on them. And we're not exactly sure what we're looking at yet, so that could be something quite exciting to uncover. Aside from that, I'm continuing my modern testbed with other species of parasites. So I've now looked at ticks, like I mentioned, but I'm also going to now look at fleas and lice that are no more than 10 years old, just to get a basis of how differently do they reflect on isotopes, and is there a huge disparity between them, or can we treat them as one and the same? And I think that'll be me very busy for the next half a year.
If I've done my job properly, you now know how and why Gaia studies arthropod ectoparasites with the aim of understanding past human and animal life histories. Hopefully that sentence no longer leaves you scratching your head for any reason other than because you're absolutely convinced that your scalp is crawling with head lice right now. I was going to say I'm sorry that I did that to you, but all of the researching and writing for this project genuinely made my head itch constantly. So I've decided it's only fair to make you all suffer too. In all seriousness, this project has been the product of weeks of work, and if you're still watching, you'll appreciate that it's only worth it if people like you support the channel. please consider sharing this video with literally anyone you think will find it interesting, and click the subscribe button so I can hopefully make videos like this full time one day. I'd like to thank Gaia for all of her hard work and in the time she put into helping me with this video. Once again, please like, comment and subscribe, and I'll see you all in the next video. Peace!